will hear a talk from Professor Sugahara and two of his students on health functions of Indonesian local crops, insight from biomolecular perspective. Professor Sugahara is a faculty of agriculture at Ehime University, Japan. He completed his doctoral program in food science and food technology from Kyushu University, Japan, and is now active as a teaching professor and a researcher for Ehime University, Japan. He is currently doing research on the enhancement of monoclonal antibody production by human hybridomas, the development of large-scale animal cell culture system, and the development of drug delivery system and novel gene factor using lipid vesicles. Professor Sugahara's recent research in Indonesia was on functional food issue in relation to Indonesian local crops, and he has been invited to a number of local and international seminars as guest speaker and keynote speaker. Before we start our power talk, we'd like to inform you a couple of important announcements. Please kindly spend some time to fill in our evaluation form after the talk to help us improve and serve you better. i3L provides scholarships for students whose parents work in pharmaceutical industry, and for more information, you are welcome to contact our marketing department. Firstly, we would like to invite our Deputy Head of Biomedicine and Head of Department of our upcoming pharmacy program, Dr. Richard Stutejo, to say a few words and welcome our honorable guest. Please give a round of applause for Dr. Richard Stutejo.
Good afternoon, everyone. So my name is not Richard. So please get the spelling correct, yeah? So a very warm welcome to Professor Takuya Sugahara. Yeah, so all the research is indeed very important for the Indonesian biodiversity perspective. Now I would like to welcome you all to the i3L, Indonesia International Institute for Life Science. So one of our concepts here is the triple helix. So we want to share between the government, the academia, and the industry. So all of these will come together to improve Indonesian overall. So one of this i3L commitment, yeah, to complete this triple helix is the power talk. Yeah. So we are very honored to get Professor Takuya Sugahara to share about this. Now currently we know we have the second largest biodiversity in the world. Yeah, do you know that? Food science, do you know that? We are the second largest land biodiversity in the world. But unfortunately, because we are falling behind, yeah, from the more advanced countries, yeah. So all of these elements of the biodiversity is not being discovered. That is why i3L, yeah, as some element to complete sort of this element to fulfill these missions, to fulfill these triple helix. Yeah, we need to look at the local crops. If professor from Japan is interested in Indonesian crops, why don't you? Do you agree with that? Yes. So, yeah, let us hear from the venerable Professor Takuya Sugahara Sensei. Yeah. So, without further ado, let's welcome Professor.
Sugahara from Ehime University, and I'm glad it's my honor to have the opportunity to study the Indonesian local crops of herds. And then at first 30 minutes, I will keep the talk about the Indonesian local crops. It has function of the Indonesian local crops, and the other 30 minutes. I come here with my students, master course students, and undergraduate students. So they had introduced study about functional food. So let's start. I'm from Japan, and do you know Ehime? You know Ehime? Ehime is here at Shikoku Island. It's the main island, Japan main island, Honshu Island. And then the Shikoku is one small island. In the Shikoku, Ehime here, Ehime Prefecture. And then we are from Matsuyama City, from here. The Shikoku is, Ehime is very famous with fisheries and agricultural, especially oranges, citrus. So one of our study is focused on the immunological function or anti-allergy function of the citrus scale excellence. But however, that I introduce Indonesian crops. So this is today's story. The one introduction and the two immunostimulative effect of allergies, and chapter 3, the immunostimulative activity of the background. And allergies is a very special cross in Indonesian background. So, and the other one is the bank of viral extracts on macrophage in vitro and in vivo. We conducted a study by using of the animal cell culture technique and then animal experiments. And then finally, we conducted human trials problem with School of Medicine at University. So let's start the story. We are very interested in the immunological function of the food stuffs. So immunity is the ability of recognition and defense of our body from some pathogens or invading materials or cancer in our bodies. So this is there are two immunological functions. One is the immune function, prevention of infectious diseases or cancers. The other one is the immune response, involves production of immune substances against specific antigens and pathogens. So screening and discovery of the potential local crops in the region to activate our immunological function, immunological skill to prevent our body from the pathogens invading materials or cancers. So we screen the immunostimulative factors from food stuffs, and then we finally developed a functional food or supplement to prevent the cancers and some other diseases. Especially allergy is very serious problem in Japan. Maybe in region also. Allergy is a very serious problem. So we are studying about anti-allergy food stuff to prevent prognosis of something, prognosis or active diseases. So finally, we will develop the functional foods possessing immunostimulative effects, and then stimulate the activity of our immunological function. So evaluation, the objective of this study is evaluation of the immunological features and the mode of action of the major local crops on immune system to facilitate the development of functional food industry in Indonesia. So first we start from the immunostimulative function of the arrowroot extract in vitro and in vivo. As you know, the arrowroot is a very common food in Indonesia. Arrowroot in a lot of food in Indonesia, and arrowroot sugar contains plenty of starch and energy source and other compounds. In Indonesia, arrowroot flour and starch are commonly used in make some food products like cakes, cookies, and chips, and etc. However, that no one knows about the immunological function as a function of food. Observed on extract at 121 degrees Celsius in hot water. The heat extracts in water stimulated IgM production by IgM process. This means that heat extracted arrowroot stimulate immunological function of our immune cells. The effect of arrowroot cells on IgM production by mice sprinklers. We collect the spermicide from the mice screen, and then put the arrowroot heat extracted sample to the culture media of the spermicide, mice spermicides. So as shown in this figure, arrowroot extract very clearly stimulate the IgG, IgM, IgA production by spermicides. So our excess obviously stimulated the Ig production, immunoglobulin production by myosin. So we examined other immunological protein. For example, interferon gamma production was examined. By interferon gamma production by myosin was examined. So arrowroot heat extracts stimulate very strongly, stimulate interferon gamma production by spirochetes. So not only Ig but also interferon gamma, other kind of the immunological protein, cytokines also production, also stimulated by arrowroot heat extracts. Then we conduct the animal experiment. We use the mice, and then arrowroot powder was administered, mixed with the feed diet, and then we 2 weeks. 2 weeks later, we collect the sample serum to examine the immunological function or administration of the arrowroot powders. We use the powders. As a result, serum Ig level obviously stimulated by the arrowroot administration. IgG, IgM, IgA level in serum was obviously activated, enhanced by the arrowroot powder administration. Now this figure suggests that arrowroot diet obviously stimulate serum Ig levels in mice. So, conclusion of chapter 1, chapter 2, arrowroot extract in hot water enhance Ig production, IgM production by Ig cells, and arrowroot extracts obviously accelerate Ig and interferon gamma production by mice in vitro. And in vivo experiment, arrowroot diet also enhanced the serum Ig levels. So this we have unfortunately we have not identified the active substance in arrowroot. However, arrowroot contain which solubilized at the high temperature have the stimulatory effect on my immune system. The arrowroot diet may have beneficial effect on our health by preventing infectious diseases. Okay, so I don't have much time, so you can proceed the next topic, background, illustrate the activity of background, 5 pictures. So background is another famous agricultural crops in Indonesia. So we prepare the background extracts. Fresh background suspended in the distilled water, conclude fiber, prepare like this, and then soak in 80% ethanol at 60 degrees Celsius for 30 minutes, filtrated and squeezed, and then fiber concentrated, oven, dried and grounded. This is the background fiber powder. This background fiber powder was provided by the manufacturers, and we used this fiber. And then we used the background fiber powder for the sample. Then background fiber powder was stirred in water for 25 degrees for 2 hours, and then centrifuged, and then supernatant was collected as BFE1. The other one is stirred for 30 minutes and heated at 100 degrees Celsius and for 20 minutes, and heat extracted. This means the heat extracted and centrifuge and supernatant as BFE2. We use these two samples. So the scheme is the same as analog. We use the i3L cells and culture, and culture supernatant was collected and to quantify the IgM concentration in culture media. And certificate was used for the gene expression levels using real-time RT-PCR method. The other one, my experience side was collected from the mice and cultured for 24 hours, and the supernatant was collected, and quantification of Igs and cytokines was examined by the analyst. This is the effect of BFE on IgG production by active cells. BFE2 heat extraction, heat extracted bank strongly stimulate IgG production by HB cells. At this point, the BFE2 stimulates gene exhibition level in HB cells to activate IgM production. So, BFE2 stimulate the IgM gene exhibition level. So, as a result of the stimulation of the gene expression level, IgM production was enhanced by the BFE2. So, BFE heat extracted backbone fiber stimulate the IgM production by T4 cells in those membrane, and it says that active substance in BFE is heat soluble and heat stable substances. So this is the same kind of experiment effect on mice spherocytes. IgG, IgM, IgA production, all these antibody production were stimulated by the BFE2, heat extracted BFE. So, cytokine production by spherocytes was examined. Interleukin 6, tumor necrosis factor alpha, interleukin 10, interleukin 4, interleukin 5, interleukin gamma production was examined. So all these cytokines production were stimulated by the backbone fiber extract, very strongly stimulated. So next we conduct the animal experiment. Mice were administered 2 doses, 27 milligram per kilogram body weight per day, and the other one lower dose was 6.75 milligram per kilogram body weight per day. So we used these 2 doses. And then 2 weeks oral administration for 2 weeks. And then finally, the serum, pile patch, and mesenteric lymph node, FLN, and spleen was collected. And then we collected the lymphocytes from these immunological tissues. And then in ex vivo assay, we evaluate the immunological function of BFE in vivo. So this slide shows the oral administration, especially high dose, stimulate the serum IgG level, IgM, IgR level in mice. And then spleen sites, IgG, IgM, IgF also stimulated. So this intake of BFE enhanced serum IgG level in vivo. So oral administration activate the immunology to enhance serum IgG levels. And BFE, intake of BFE strongly stimulate the splenocytes, as shown in these figures. So why? This is the mode of action, one of the mode of action of the BFE on lymphocytes, nice lymphocytes, orally administered lymphocytes. We collect the lymphocytes from the spleen. Then we examine the gene expression level of IgG1, IgM, IgA. So as shown in this figure, spleen lymphocytes and patch lymphocytes strongly stimulated the gene expression level of these antibodies. So background fiber extracts stimulate the gene expression level of the lymphocytes from spleen and patch, and then finally activate the production level of these antibodies. This is the cytokine production effect of BFE on cytokine production by lymphocytes in vivo. So interleukin 4, 6, 10 very strongly stimulated, shown in red color characters. So interferon TNF alpha, TGF beta, interferon gamma also very strongly stimulated, especially spleen production levels. And MNL lymphocyte production level is strongly stimulated by the backbone oral administration. Spleen, MLM, patch lymphocytes stimulated by the oral administered BFE. And then this slide shows the intake of BFE maybe activate our immunological cells and then prevent the embedding of the pathogens. Then protect our body from the maybe cancer also. So BFE stimulated the IG production by IG cells and also activate the mice lymphocytes, IG mice lymphocytes. That oral administration activate the IG levels and the cytokine levels strongly activated by the oral administration of BFE. So BFE may activate the innate immune response by enhancing macrophages. So background intake may have a stimulatory effect on the immune system and then contribute to our health. So next, the same sample, background fiber extracts, effect of background fiber extracts on macrophages. Macrophages participate in the innate immune system. This is naturally, we have the natural immunological functions. It's independent for the antigens. So macrophage is very key immunological cells to activate the acquired immune system. Acquired immune system is the antigen-dependent immune system. So macrophage is the key cells between the innate immune system and the acquired immune systems. So activation of the macrophage is very strongly activate our acquired immune systems. So we examine the effect of fiber extracts on macrophages. The macrophage is here. Macrophage has the key player of the essential and prototypical role in innate and adaptive immune systems, acquired and adaptive immune system against infection by phagocytic activity and degrading pathogens and dying cells. The activated macrophages are considered to be associated with cytokines such as interleukin 1, interleukin 6, TNF alpha to recruit and activate other cells to initiate adaptive responses. So evaluation of the stimulatory effects of background fiber exercise on macrophage function is very important to investigate food functions. So as a result, we use cell culture, animal cell culture technique. We use J774 cells, macrophage cell line from mice. TNF-alpha interleukin-6 production, those dependently stimulated by the BFE. Gene expression level also stimulated. Then activation of the gene expression level is resulted in the high production levels by J774 cells. This slide showed this slide showed the phagocytotic activity. Phagocytosis is the eating activity of the macrophage, eating the pathogens, bacteria infected in our body. This means phagocytosis. So BFE very strongly activate the phagocytotic activity of macrophage. So BFE activates macrophage to eat pathogens embedded in our bodies. Very strongly activated by the macrophage actions. So phagocytosis is very important function of macrophage. So macrophage can be activated by BFE for cytokine production, such as TNF-alpha and interleukin-6, and then also activate BFE, activate the phagocytosis, eating activity of the macrophage. So this is a result of the in vivo trial. Mice were administered 2 levels of the bank fiber exercise, 26.8 milligram per gram body weight per day, and the high dose is was 27 milligram per gram body weight per day. Especially a high dose administration, bank of fiber exercise very strongly activate the PMAC. PMAC is peritoneal macrophages. So oral administration of BFE facilitates the interleukin-6 and TNF-alpha production of peritoneal macrophage PMAC in vivo through the facilitating the transcription activity. So BFE activates the transcription activity of PMAC and then resulted in the high production level, protein production level of macrophage was achieved by the BFE administration. So phagocytotic activity also strongly stimulated by the bank of fiber administration. So cytokine production and phagocytotic activity also stimulated by the orally administered bank of fibers. So I need to hurry up. We tried the identification of the active substance in bank of fiber, pectinase. The bank of fiber exercise, biological function of BFE is very stable. So we suppose the active substance is maybe carbohydrates. So we use the pectinase to destroy the carbohydrate or in BFE. After pectinase treatment, bank of fiber extract biological activity, immunological stimulation activity of the bank of fiber was obviously decreased by the pextinase treatment. IL-6, especially IL-6 products, but TNF products also are suppressed. Then pextinase treatment partially inactivate the effect of BF3 on macrophage cells. This was that one, but maybe BF3 contains some active substances, and one of the active substances contained in BF3 is pexin. Pexin is dietary fiber. Water soluble dietary fiber is very stable, and then maybe one of the active substances, pexin, and the pexin activate the immunological cells. Pexin from bank fiber activate our immunological cells to prevent infectious diseases. So arrowroot and bank from Indonesia have immunostimulatory activity. Stimulate the lymphocytes and stimulate the macrophage. Then they could be developed as a functional food with potential to modulate the immune system and health function. They are expected to contribute to the food industry in Indonesia based on the local crops. Yeah. So finally, it's just the arrowroot and bankworm are very normal, very common agricultural crops in Indonesia have some our health function of our bodies. Okay, this study was conducted by the Indonesian my Indonesian student. Doctor, a PhD student. She already got a PhD and have a job in Indonesia. And this is my collaborator in Indonesia at the Japanese University. And Alex also my collaborators. And then these are my faculty laboratory staff. And I'm in the laboratory and technology. And then these are my students. Thank you very much. And then we need to meet to my students. So I would like to move to the presentation of my students. But first, Nanami Kurihiro. She is an undergraduate student of my laboratory. She is using Japanese traditional food stuff, anti-allergy, immunology effect of the Japanese traditional food.
My name is Nanami Kunihiro. I am a postgrad student in Ehime University. I am very happy to be able to talk to you. Today, I'd like to talk about immunostimulatory effects of dried bonnet extract on macrophages. First of all, let me talk about dried bonnet. Dried bonnet is one of the most popular preserved foods in Japan. It is well known that dried bonnet is rich in inosinic acid, which is a good taste umami substance. Dried bonnet also has various health functions, including bloodstream improvement, anti-aging, fatigue recovery, and so on. The powder of dried bonnet emerged during shaving process is normally discarded. However, dried bonnet powder is valuable. To develop dried bonnet powder for functional foodstuff, the health function of dried bonnet powder is evaluated. Previously, it has been shown that dried bonnet extract stimulates immunoglobulin production, by human lymphocytes and mouse spleen lymphocytes. However, it is not clear the effect of dried bonnet on macrophages. Therefore, the evaluation of stimulatory activity of macrophage was conducted. Overview of the immune system is summarized in this slide. There are two types of immune responses, namely innate and adaptive immunities. Innate immunity is antigen non-specific and provides immediate defense against infection. On the other hand, adaptive immunity is antigen-specific and composed of highly specialized cells and processes that eliminate or prevent invading pathogens. When antigen enter our body, firstly, innate immunity is activated. Macrophages play a critical role in innate immunity and also activate adaptive immunity. This slide shows the introduction of macrophage. Macrophage produces immunological factors such as cytokines and chemokines. Macrophage also produces nitric oxide to eliminate invading bacteria. In addition, macrophage engulf and digest foreign substance, microbes, and anything else. This process is called phagocytosis. The adaptive immune After digestion of pathogen, macrophage presents the pathogen as antigen on cell surface. The adaptive immune system is activated this antigen presentation by macrophages. This slide shows the method of sample preparation. Dried bonnet extract, abbreviated as DBE, was prepared by heating of dried bonnet powder in water for 2 hours. After centrifugation, the supernatant was collected and dialysis with dialysis membrane of molecular weight cutoff of 14 kilodalton. Mouse macrophage-like cell line J74.1 cells and RAW264.7 cells and mouse peritoneal macrophages (PMAC) were used in this study. The activity of macrophage was evaluated by cytokine production, gene expression, signal activation, and phagocytotic activity. Firstly, the effect of TBE on inflammatory cytokine production was evaluated. J74.1 cells were cultured in the medium containing TBE for 6 hours, and TNF-alpha and IL-6 in the medium was determined by ELISA. As a result, TNF-alpha and IL-6 production was activated by TBE in a dose-dependent manner. Next, the effect of TBE on messenger RNA expression levels of TNF-alpha and IL-6 in J74.1 cells. TBE was added to the culture media activate the transcription of inflammatory cytokines. On the other hand, in MAP kinase signaling pathway, kinase cascade is phosphorylated by MAP kinase being phosphorylated. Then ERK, JNK, and T38 are phosphorylated and activate to examine the consignment of kappa B signaling pathway in the regulation of cytokine production. J7 4.1 cells were cultured in the medium containing TBE for 16 minutes, and the protein level of signal molecules were determined by Western blot analysis. As a result, TBE stimulates phosphorylation level of I-kappa B alpha and nuclear translocation of N-kappa B. In addition, the effect of TBE on MAP kinase signaling pathway in J7 4.1 cells were also examined. As shown in this figure, TBE did not stimulate phosphorylation of ERK, even though TBE enhanced phosphorylation level of JNK and T38. Those findings suggested that TBE stimulates cytokine production through activation of N-kappa B and MAP kinase signaling pathway in macrophages. It is well known that LPS binds to toll-like receptor 4 on cell surface of macrophage and activates cytokine production. To examine the involvement of TLR4, the effects of TBE on cytokine production under the TLR4 inhibited condition was examined. As a result, inhibition of TLR4 signaling significantly cancels the effect of LPS on macrophage activity. However, the effect of TBE on cytokine production by J7 4.1 cells was not affected by TLR4 inhibitor. Next, the effect of TLR4 inhibition of N-kappa B signaling pathway, N-kappa B transduction induced by TBE was examined. As a result, TBE still enhanced nuclear translocation of N-kappa B. And those findings suggested that TLR4 signaling is not dominant for macrophage activation by TBE. Next, one of the important functions of macrophage is phagocytosis. Therefore, the effect of TBE on phagocytic activity was evaluated by flow cytometry. As you can see in this figure, TBE enhanced phagocytic activity at 2%, whereas it was 36%. Next, the effect of TBE on phagocytic mice or TBE. After 2 weeks of oral administration, macrophages were collected and the effect of TBE on phagocytic activity was evaluated. And 4.1 cells and PMAX. TBE also significantly enhanced the messenger RNA expression levels of these cytokines in both cells. In addition, TBE also activates N-kappa B and MAP kinase signaling pathway. These facts suggested that DBE facilitates cytokine production through activation of N-kappa-B and MAP kinase signaling pathways in macrophages. Furthermore, DBE activated the LR4 inhibited J1741 cells. It was suggested that DBE acts on not only TLR4 but also another receptors. And DBE activated the phagocytic activity. From those findings, it was suggested that DBE contributes to activate phosphodefense against infection. Thank you very much for your kind attention.
So we will put the student master student next 15 minutes.
Good afternoon. First of all, I introduce myself. My name is Teja. Student in Ehime University. It is my pleasure to have a chance to talk about our recent study of the novel function small dry sardine. First of all, I'd like to start my presentation from introduction about type of allergic response. Allergic disorders such as pollinosis, food allergy, and rheumatism are increasing problems all over the world. More than 25% of people have the allergic symptoms in the world. Allergic symptoms can be categorized in 4 types: type 1, type 2, type 3, and type 4. We focus on type 1 allergic symptoms because it is the most common and serious problem for us. In this slide, type 1 allergic response is summarized. For example, pollinosis. When allergen invades our body through the nose, eye, or mouth, allergen is processed by dendritic cells. In lymph nodes, dendritic cells induce differentiation of naive T cells into Th2 cells. Subsequently, Th2 cells secrete interleukin-4, which activates maturation of B cells into the IgE-producing plasma cells. IgE binds to mast cells through the high-affinity immunoglobulin E receptor, Fc-R1. In the case of second attack by allergen binding to the IgE on mast cells, it triggers the release of granules containing chemical mediators such as histamine, leukotriene. This process is called degranulation. It is important to inhibit the degranulation for reduction allergic symptoms. In this slide, I'd like to show you the importance of anti-allergic treatment. Anti-allergic medicine were usually used for reduction allergic symptoms. However, sometimes serious side effects such as sleepy or thirsty are problem. On the other hand, side effects of anti-allergic treatment can be neglected. So, anti-allergic treatments are more safer than medicine. In addition, we can take functional food in daily life. We did the screening, Therefore, anti-allergic food is effective to prevent allergic symptoms. We did the screen test about anti-allergic effect, and we found that small dry sardine extract has an anti-allergic effect. In this presentation, anti-allergic effect of small dry sardine extract will be discussed. This is small dry sardine. Small dry sardine is a traditional food stock in Japan, normally used for making broth, which is the base of Japanese cuisine. In addition, small dry sardine is rich in calcium and with nutrients. Therefore, small dry sardine is a traditional food stock in Japan. However, there are no studies on the about anti-allergic effect on the small dry sardine. Therefore, in this study, small dry sardine extract was evaluated, and the model and used in this study. Small dry sardine is digested with protease peptide to make water-soluble extracts. And protease digested small dry sardine is abbreviated TSDS. For degranulation assay, we used rat-specific leukemia cell line RBL283 cells. RBL283 cells was cultured 5% FBSD and EM. Degranulation is assessed by beta-hexane dense release assay. In this slide shows sample preparation methods. ESDS extract centrifuged and its supernatant was collected and dialyzed with dialysis membrane molecular weight of 500 Dalton to eliminate the salts. Then it used for degranulation assay. In this slide, beta-hexane dense release assay is summarized. ESDS extract added to the culture medium of the Ig sensitized RBL283 cells. Degranulation was induced by addition antigen. The release of beta-hexane dense was determined as a marker of degranulation. In this slide shows the results. The effect of ESDS extract on the degranulation and survivability of RBL283 cells. As a result of beta-hexane dense release assay, ESDS extract inhibited the degranulation in a dose-dependent manner. In addition, the cell viability of RBL283 cell was not affected by addition of ESDS extract at any dose. From these results suggest ESDS extract suppressed the degranulation without side toxicity. To examine the molecular characteristics of the active substance, the effect of dialyzed ESDS extract on degranulation was investigated. As you can see this figure, ESDS extract dialyzed with molecular weight of 500 Dalton. 500 Dalton suppressed the degranulation. On the other hand, ESDS extract dialyzed molecular weight of 14,000 Dalton did not suppress the degranulation. It is supposed from this result, the molecular weight of the active substance in ESDS extract is between 500 and 14,000 Dalton. In addition, to reveal the molecular characteristics of the active substance in ESDS extract is heated at 100 degrees Celsius and 14 minutes. And remaining activity was evaluated. As you can see this figure, the activity was not affected by heat treatment. Therefore, the active substance in ESDS extract is heat stable. In this slide, signaling pathway involved in degranulation is summarized. In IgE-mediated allergic symptoms, initial signal is provoked by cross-linkage antigen with IgE bound to Fc on R1. Degranulation signaling pathway is divided to two processes. One is calcium ion dependent pathway. Another one is calcium independent pathway. Microtubule formation and granule translocation is in this process, and subsequently degranulation will occur. The elevation of intercellular calcium ion is a critical process in the degranulation. Therefore, the effect of ESDS extract on the elevation of intercellular calcium ion was examined. As you can see this figure, intercellular calcium ion rapidly increased after stimulation the antigen. However, the elevation of intercellular calcium ion was not decreased in the presence of ESDS extract. It is supposed from this result the anti-degranulation effect of ESDS extract was not accompanied with the elevation of calcium ion. In addition, the degranulation suppressive activity of ESDS extract was examined under the stimulation of calcium ionopathy that carries calcium ion into the cytosol across the plasma membrane. As a result, even though ESDS extract inhibited the degranulation, intercellular calcium ion was not decreased. Therefore, it is supposed from these results, ESDS extract inhibited the degranulation in a calcium ion independent manner, and ESDS extract inhibited the degranulation at downstream of the calcium ion elevation step. Therefore, the effect of ESDS extract on the downstream of calcium ion elevation step, but namely microtubule formation was examined. In this slide shows the effect of ESDS extract on microtubule formation in RBL2 cell was examined by immunofluorescence microscopy. Microtubule formation is key process in degranulation. As you can see this picture, intact cells did not spread out microtubule. On the other hand, as you can see this picture, antigen stimulation induced microtubule to spread out. However, ESDS extract completely suppressed to spread out microtubule. It is supposed from this result, ESDS extract inhibited the microtubule formation to suppress the degranulation. Here, the effect of ESDS extract on the passive cutaneous PCM mice was examined. If histamine is released by degranulation, blood vessel permeability is enhanced. Therefore, the effect of ESDS extract on the degranulation in vivo was examined this PCA model mice. PCA reaction was induced of IgE into ears, and after 24 hours later, antigen and Evans glue was injected into the tail vein. Finally, the dye was extracted from each ear, and absorbance was measured. As a result, PCA reaction was significantly suppressed by oral administration of ESDS extract. It is supposed from this result, ESDS extract suppressed the degranulation in vivo. In this slide shows the effect of ESDS extract on the cellular polynosis model mice. Polynosis is induced the internal injection of cellular pollen allergen at 11 times during 5 weeks. And other symptoms such as sneezing and rubbing was observed. As a result, rubbing frequency, sneezing frequency is suppressed administration of ESDS extract, especially sneezing frequency significantly suppressed. It is study will reveal the anti-allergic effect of small dry ESDS extract in vitro and in vivo. Therefore, we found small dry sardine has the potential to be anti-allergic. Thank you for your kind attention.
Thank you for your insightful talks today. We'd like to invite is there any question from anyone?
Well, thank you for the presentation. It was a really good studies. Well, my name is Pulomas. I'm from Biomanufacturing Training Program. I have a question about the two presentations that you made. It's about, do you think there is any possibility and the bonito have a toxicity effect into other cells?
Toxicity?
Yeah, toxicity effects.
We already checked about the toxicity. They're not toxic.
Into another cells.
Yeah.
Because in the results that you have already given us,
yeah.
The trend is when you give a higher concentration, it gives also the higher effects too, right?
Yeah.
So in order to balance anything in the body, do you think?
Yeah, of course.
So there's
too much the top, some kind of toxic, but we will find out the optimal concentrations to activate the neurological function. But actually, not toxic, but too much high concentration. Anything is toxic, right? So this is not actually this is not this substance of food. So we take these substances as food, the non-toxic, as well as the extracts also not toxic, but too much is toxic.
Is there any other question? Yeah.
Okay, so Professor, because you are mentioning about this this kind of plant and have this potential immunity effect, I'm wondering if you have any by you or by any other research that has been done, by consuming this food regularly may prevent some of the very common diseases like in Indonesia, or maybe improving immune for some of the certain sickness. Are there any report on that by eating the arrowroot and bone and also the body? Are there any other reports about clinical science?
You mean
so let's say that I'm eating bengkuang regularly and probably in the mouse or maybe in the person. By eating that regularly, some of the disease, has been shown to be diminished. Are there any other? investigation of this?
Yeah, this case of these first stuff have the evolution functions, but we really in it. We also we have the monitor and so it's
Sorry, the reason why I'm asking because bengkuang is one of the most common mask. I mean, I'm sure the Indonesian women like to use bengkuang to suppress a lot of the skin-related diseases. So I'm seeing this probably there are some connections with those.
yeah.
Do you agree with that?
Yes. Yeah, I don't have those ideas of that. But considering the background, when the background is the popular and it's usually like cosmetics. So the fiber extracts, maybe one of the active substances pectin. The pectin consuming is big. Pectin has many of the biological functions, such as reducing cholesterol and reducing blood pressure. We are shown here with biological functions. So, and then maybe it should be good for our skin health. And we can consuming the background, and then we protect our control and be healthy of the whole body. Yes.
Do we have one more question? Oh, three more, okay.
Do you think the ESES extra can also help balancing the TH1 and TH2 levels? Here the mechanism of ESES extra is it depends degranulation, right?
Yeah.
So ESES extra can also help to balance the level of TH1 and TH2 because in allergic people the immune system is skewed towards TH2 cell, right?
Yeah. It is important to what you say, the TH1 and TH2 balance is important to prevent the allergic symptoms. But we focus on just the mast cells about degranulation. It is about TH2 and TH1 balance, but it is not researched. So it is that it is I want to continue to experiment about your advice. Yes, thank you.
I guess the pH balance is very important for the IgE product in your body. Just keep shown is after IgE production, after IgE production, this phenomenon is that produced IgE is combined with base pair substance, and then after this substance suppress the just base pairs. Maybe it don't matter with the TH balance. TH balance is very important. The whole process of the yeast family. Okay.
Other questions? Everyone?
I'm just consider in regard to the diet of the arrowroots that you give to the mice, it's around how many grams per day?
I showed the.
And yeah, I mean, like after that, I would like to, let's say, substitute or convert this diet to the mice into the diet of the human. So let's say like how many gram average that, let's say, the human diet could have the benefit of eating arrowroot powder every day. Because now in Indonesia, I think arrowroot also already well known due to its low glycemic index. So it's been used widely for flour wheat substitute.
So you mean how much we need to eat an arrowroot?
Yeah, because in the mice, I saw it in your research. I think it's how many grams?
Actually, we didn't check about the point. We just put the mass diet of the arrowroot powder, but let's see the.
There
Arrowroot in the.
So almost more than half kg.
Our powder is 600 from our diet. So according to from this data, it's just actually we don't check about how much we need to eat.
Yes.
But supposing from this result, maybe we need arrowroot, maybe some kind of